Dear Professor Hershko, it's a pleasure to have you among us. We are at SIPS 2022 at the plenary session, and we welcome you with your very interesting presentation. Please, your podium and the screen is yours.
Thank you very much, Florian. I am very happy to talk to you. Unfortunately, I couldn't come, but at least this way. And I wanted to share with you some of the thoughts that I have had about the importance of basic research for innovation in industry. Because as you know, as you all know, one of the most important things in industry is innovation. Then you get ahead. So my background is from medicine. I studied medicine many years ago, about 50 years ago. And then I got interested in biomedical sciences. So that's what I know most about, and that's what I talk about, but it has general implications for all sciences and all kinds of innovations in science. So when I studied medicine about 50 years ago, and when I compare it now, how medicine is today, there are some huge differences. We can see some of these differences or advances in the first slide. Can I have the first slide, please?
Yeah, sure. Just a moment.
Yes. So when we look what happened in the last 50 years, there was a large increase in life expectancy, more than 10 years. And it's not only life expectancy, also life quality. So medicine has provided not only longer lives, but also longer, more meaningful lives. And that was due to great improvements in the many things. One was great improvement in the prevention of disease. For example, in cardiovascular diseases, many risk factors have been treated, such as the lowering of cholesterol by the use of statins, better hypertension management by a variety of effective antihypertensive drugs, which, of course, greatly improve the cardiovascular situation. The recognition of a healthy lifestyle, that's quite recent. 50 years ago, we didn't know that the very simple things that we can do, like certain diets, exercise, and so on, that can improve our cardiovascular system. So that all these improve the prevention of disease. Also, there were great improvements in the diagnosis and early detection, which is very important. There was great improvement in imaging. 50 years ago, when I studied medicine, there was no computerized tomography, no MRI, not even ultrasound. All these are very important now for diagnosis, for early detection, which is so important for medicine. And that was mostly a technological improvement. So the improvements in technology also greatly improved. Innovations in technology greatly improved medicine. And there are many others, like molecular diagnosis, like the PCR that we are using today for diagnosis of coronavirus. All this was not there. And there were also great improvements in the treatment of disease. Once the disease was there, it could be, can I have the slide back, please?
Sure.
Mm-hmm. Yeah, there were great improvements. Some of the in the treatments, some were technology treated, like pacemakers that we didn't have then, and well, now it's very common. Intensive cares, which greatly improved many acute situations. And there are also a great variety of new drugs, novel drugs, such as in cancer therapy, targeted treatment of cancers. AIDS has been converted from acute fulminant disease to a chronic treatable disease. And of course, personalized medicine is a great improvement, recent improvement. So all these great changes that we witnessed were due to scientific discovery and the use of these scientific discoveries for innovations in healthcare. And what I want to point out in this, I will try to tell you in this lecture is that basic science and discovery provides the foundation for innovations that resulted in great advances in medical care, and that will continue like that. We still have great challenges, and further advances also need further basic research and discovery. Applied research and development are important, and they are based on discoveries of basic research. So they benefit healthcare directly and in the short term, but in the long term, they require the basis of basic research. So number three is that applied and basic research can be combined, but applied research alone without the basis of basic research cannot go very far. So these are the things that I would like to show you, and here are some examples. As you will see in the next slide, please. These are Watson and Crick with their model of DNA. That was the discovery of how our genetic material, what is the structure of genetic material. And that was a purely basic research. What they wanted to know, Watson and Crick, are what is the molecular basis of heredity. As in all basic research, they wanted to know how things work. They were driven by curiosity. So basic research, one of the main things in basic research is driven by curiosity. But later on, if you fulfill your curiosity, you also help mankind, and you will see it now. When Watson and Crick discovered the structure of DNA, they did not think about application. But this discovery had a huge impact on biology and medicine, and there were many, many, many applications. For example, precision or personalized medicine is based on DNA technology now. Genetic diagnosis, by which we can diagnose very early all kinds of genetic problems, is based, of course, on DNA diagnosis. The formation of recombinant proteins like insulin, for which there is a huge market of hundreds of millions of people who suffer from diabetes all around the world. It's produced by recombinant technologies, which is based on DNA technology, and so on and so forth. So the DNA technology is very important in health and in many other things, and it was very important in a huge number of innovations in healthcare. As we will see in the next slide, here's another example. This is Alexander Fleming, who discovered by penicillin, the first antibiotic. And that was done by curiosity, by chance observation. One day, Fleming saw that there was a contamination in the dish in which he grew his bacteria. And it was a contamination by fungus. And he noted that around the fungus, there was an idea where the bacteria died. So he thought maybe the fungus secretes some substance that kills the bacteria. And that substance was later isolated by others. It turned out to be penicillin, and when it was produced, it saved millions and millions of lives. It also showed the way for the discovery of other antibiotics and just changed the therapy of infectious diseases. So here again, a basic discovery led by curiosity, which had a huge impact on innovation in medicine and in health-related industries. Still another example, as we'll see in the next slide, is the discovery of reversible phosphorylation of proteins by Fisher and Krebs, two biochemists, who were interested in finding out how the activities of proteins are regulated in cells. It was a big research, and they found it is done by enzymes called the protein kinases, and this resulted in the development of protein kinase inhibitors in the targeted treatment of cancer. The first one was Gleevec, which was more than 20 years ago. It was the first kinase inhibitor. That was the first targeted treatment of a leukemia, of the blood cancer, and it caused wonderful remissions without all the side effects that were done by the non-targeted treatments of cancer. And now there are scores. In 1918, there were already 37 FDA-approved protein kinase inhibitors that targeted many kinases for the treatment of a variety of cancers. So as you see, there are many examples, and here one more example is my own work. I am working on how proteins are degraded in cells, and I realize this is an audience, many of whom don't have a basis in biology or biomedical sciences, so I try not to be too technical and try to explain what I am doing in a language which will be understandable, I hope. So I'm working on how proteins are degraded. Now, proteins are important. The machines of our body. They carry out, we have thousands of different proteins that carry out all the chemical and physical and many other functions in our body. So we have thousands of different proteins who carry out different functions. How can we have so many proteins? Before this, we have to know how proteins are built. They are built by assembly of building blocks called amino acids. When they are joined together, they form a protein. It's like beads on a string, and the string of proteins can be a several hundred amino acid bases building blocks long. And the difference between the different proteins is the order or the sequence on which the 20 amino acids are arranged, and there can be millions of different sequences in which you can arrange 20 amino acids along a protein of, let us say, 300 or 400 amino acids. And now the sequence is what is the sequence of each protein, or what is the order of amino acids, is determined by the sequence of bases in the DNA, the genetic material. So the genetic material, it is the blueprint for the formation of proteins. The genes in the DNA tell the protein what will be the sequence, and when the sequence is formed, a certain protein will be formed. So that is an important process of protein synthesis. But when I started out to work, there was another, the opposite process was also known protein degradation, in which the proteins disassembled back to free amino acids. And not very many people were interested in protein synthesis, but not very were interested in protein degradation, because it seemed wasteful. Why? To degrade back a protein once it is synthesized, and it can do some good. But it turned out that protein degradation has important functions in the cells, as we shall see in the next slide. Next slide, please. As we can see in the next slide, please, Florian. Okay, so I asked the question, how are cellular proteins degraded at a highly selective and regulated mode? But I would like to go back to the previous slide. Yeah, so this shows the main functions of protein degradation. One function is to remove abnormal proteins. The protein, which is folded, has a very delicate structure, which can be misfolded very easily, can be damaged. When it is damaged, the abnormal protein is toxic. It can kill the cells in which they are. For example, in the disease called Parkinson's disease, there are certain cells in our brain in which abnormal proteins accumulate. Usually they don't, because it's removed, but there is some problem there. And when they are not removed, they kill the cells and cause this awful disease. And another function of protein degradation, number 2, is to degrade completely normal regulatory proteins to stop their action. So a protein acts for a certain time, but then it has to be stopped. Let us say I can liken the cell to a huge orchestra in which has thousands of different proteins, which are the players in the orchestra, and they all have to play in harmony. Let us say that we have one player, let us say a drum that drums, but then it has to be stopped. If it doesn't stop, it can ruin the symphony. So stopping is also important in biology, like it is important in the working of a symphony. So stopping of the action of proteins is done by degradation. So these are important functions, and I ask the question shown in the next slide, how are cellular proteins degraded in a highly selective and regulated mode, because a certain protein out of the thousands of proteins is degraded only when it should be degraded, and it is degraded in a way that is regulated. So I won't go into all the details, that is all very technical, but basically we found that proteins are targeted to degradation by being linked to a small protein called ubiquitin. So ubiquitin is a protein that when it is attached to a protein, it's kind of a kiss of death. It tells the machinery that it should be degraded. And as you can see here, the system that we discovered it has two main parts. One is the linkage of the ubiquitin to the protein. The protein is that orange thing into which the little blue balls called the ubiquitins are linked by the action of some enzymes that know which protein to link the ubiquitin. Usually there's also a chain of ubiquitin this way. And in the second part, that is the tagging process. In the second part, proteins that are being tagged by ubiquitin chains are degraded by a structure called the proteasome. The proteasome is like a big machine, like it's like a big barrel. In the inside, it degrades the protein. So it is like a food processor, if you want, but a very sophisticated food processor, because it will degrade or it will push inside into the cavity where degradation occurs only proteins that are linked to ubiquitin chains. And once that happens, the proteins are being chopped up to small pieces, and from there they are back to amino acids. And ubiquitin is not chopped up; it's recycled. It can be used again to degrade another molecule, another protein molecule. So that is in a nutshell how the system works. We worked on it several years until we found out all these structures. And then other investigators got into the system, and it was found that ubiquitin-mediated protein degradation, as the system is now called, has very many important functions. As basic as a cell cannot live with it, it has important functions in the living of the normal organism, of the healthy organism. For example, it controls the cell division. Our cells are divided when they should divide, and they do not divide when they should not divide. So that is under control of some proteins that have to be degraded in order to work in a regulated mode. So here again, a regulatory protein has to be degraded in order to control cell division. And signal transduction. Can I go back to the slide, please? Signal transduction, like where the cell gets signals from outside in changes in the environment, and then they respond by proper responses. The ubiquitin system is involved in that. It's involved in the inflammation and immunity by destroying some inhibitors. So that is kind of a negative control. You can set to motion a certain process by removing the inhibitor, like removing the brake on the system, and so on. So ubiquitin-mediated protein degradation has many important roles in the healthy organism. And it was found that when the ubiquitin-mediated protein degradation does not work correctly, it has important roles in disease, as we can see in the next slide, please. Next slide, please. Yeah, it was found that the ubiquitin system, malfunction of the ubiquitin system is occurring in many diseases. Many cancers, many human cancers are due to abnormalities in the degradation of regulatory proteins. Mostly proteins that stimulate cell division are not degraded, and if they are not degraded, the cell divides when it should not divide. So that's the essence of cancer. It happens in many cancers. Ubiquitin malfunction of the ubiquitin system is involved in common neurodegenerative diseases like in Parkinson's disease. As I said, some abnormal proteins accumulate where cells in the brain when they should not, and they kill these cells, and that causes the disease. Also, dementia by Alzheimer's disease is caused many times by the accumulation of abnormal proteins in other cells in the brain. So these are very important diseases, and there is a lot of research going on to try to find out how to get rid of these abnormal proteins in these neurodegenerative diseases, and so on. In mental retardation and in other diseases, ubiquitin system is involved. And because the ubiquitin system is involved in so many diseases, the pharmaceutical industry began to work on it to try to find new targets for the targeted treatment of cancers. And that is the first drug that was developed. It is a drug called Velcade or bortezomib. It inhibits the proteasome, and it is effective in the treatment of multiple myeloma. Multiple myeloma is a bone marrow cancer. These are cancer cells that grow in the bone marrow, and they destroy the bone marrow. They destroy the bone. They also destroy the function of the bone marrow, which is to make blood cells. So there is a big problem in the formation of normal blood cells. And because of that, multiple myeloma was an awful disease, very fulminant disease. But with the treatment of this proteasome inhibitor that kills the myeloma cells, there was a big change in the treatment of disease. With the treatment by Velcade and some other agents, now people can live many years of good quality life. So it doesn't cure the disease, but it converts it to a chronic disease which has good quality life for many years. So because my background is from medicine, for me it was very satisfactory to see that the basic research that I have done for many years now can be used to improve the health of many hundreds of thousands of people all around the world. And the pharmaceutical industry is now very busy to work against to try to develop other drugs that are targeted against the ubiquitin system. So Velcade was not developed by me, but it was developed based on our basic research, which discovered the ubiquitin system for protein degradation. And so once again, the benefits of investment in society, in basic research, were returned to society. So to sum up in the next slide, please, my conclusions are that basic research is the long-range investment of society for technological innovation. If we want to have highly original innovation in industry, society should support high-quality basic research. And I also would like to thank the people who helped me for many years in my lab. Many of these people who are Dwara Ganot, Heller, also my wife, Judy Hershko, helped me for many years in the lab. And I have many former graduate students, including Aaron Chahanover, who worked with me during the discovery of the ubiquitin system, and for that he shared the Nobel Prize with me. And I was also glad to share the Nobel Prize with Erwin Rose from Fox Chase Cancer Center, with whom I spent sabbatical years, and we made some part of the work in collaboration of the discovery of the ubiquitin system. These are the present lab members who help me now in finding out the roles of the ubiquitin system in the control of cell division. So thank you very much for your interest in this system.
Thank you very much, Professor Hershko. Very interesting the history of the role of basic science in the health of human beings. And this is a great, message to government and politicians that if they want the quality of life, and if we want to fight the different diseases like cancer and the others, they have to invest in basic research.
Yes, I believe so. I believe so. And the history of medical sciences shows that it is like that. And I think also in other fields, but my expertise is medical sciences. I talked about that.
uh, because we always say, since SIPS is a multidisciplinary
Uh-huh.
summit with
Yes,
50 different symposia in different fields of science, they are in the same roof. It's unique in the world. That is exactly our idea, to bring together different fields, science, medicine. So medicine is one of the strongest this year. We have three, different medical symposia. One is antioxidant, one is new technologies in medicine, where your presentation falls in, and the other one is COVID. So we are bringing those in the same roof with mathematicians, with technologists, with scientists, with advanced materials, with intelligent materials, because all they help medicine. Correct?
yes, yes. And again, I am very sorry that I couldn't come in person, but I hope that next year I'll be able to.
Sure, you are invited next year. What I wanted to just to ask you the question is, uh, so basic research. Uh, Now sometimes scientists do the basic research, but they do not apply, and you mentioned a little bit in your presentation. So, and that we are trying to achieve in order to pass the value of death, to have it applicable in the society. That's why it is sustainability through science and technology, because it's science that helps
Yes.
life.
Yeah, that's true. But I believe that even with the scientist who makes the discovery doesn't apply it, others will apply it because becomes basic knowledge of all humanity, and others can apply it. And we saw it many times. So if you make this big a great discovery in basic research, do not worry about the application. The applications will come. It will certainly will come.
Mhm.
It takes time, but they will come.
And how to shorten this distance between the basic research and the application is a way. What's your suggestion?
Well, it
It is the crystal ball.
can be short by having can be shortened by having a connection between the basic researchers and the industry, and there are all kinds of between academia and industries, and that is being done. Uh, Sometimes people try to be too short, and try to find the application right away, and usually they fail. Look at there are many examples in history. Maybe you are too young, but there was the war on cancer that was declared by President Nixon, and he gave a lot of money on it to fight cancer, to find a cure in 3 years. Nothing came out of it. That's not how you cure cancer. You should try to find out the basics, and once the basics are there, then it can be translated. So if you try to translate too early, that's not very good. But of course there should be connection between academia and the industry, and sometimes some very good things come out of it.
Yes, thank you, because this is what SIPS is about. We bring them all together.
Yes,
That's why your presentation is so relevant to our SIPs. So basically, it is a confirmation of what we are doing. So I thank you very much for this excellent presentation. So you are right where we are targeting exactly the importance of basic science, but also through the application in order to serve the society. And you mentioned so many examples of extending the human life, turning cancer from a deadly disease to a chronic disease, and also sometimes even curing it. So really, we thank you for this presentation, and you are already invited in SIPS 2023 for another enlightening presentation like this one.
thank you and good luck in your conference.
Thank you also. Thank you.
Okay,
We look forward for the next year. Thank
okay,
you.
bye bye.
Thank you.